So callus growth has at least three phases. First of all, the induction phase. Second, the rapid division and the growth phase, more or less this is like a log phase. And then ultimately there would be a declining phase where the callus is not anymore growing and there could be infection in the callus and due to the scarcity of media and nutrients. So plant hormones are used to induce or initiate the callus formation. So let us look at the factors that induce callus formation. Few factors which are essential for callus formation is auxin and cytokinin. The relative balance of auxin and cytokinin determines few factors in terms of plant growth. If auxin is high, cytokinin is low, then root development is augmented. If cytokinin is high, auxin is low, shoot development is preferred. But if auxin, cytokinin are in a equivalent 1:1 ratio, then callus development is induced. Let us talk about the advantages and disadvantages of this callus culture. The advantage includes making transgenic crop, reviving infected crop that we have learned from many stem cultures, and plant biomedical researches. Ultimately, disadvantage includes the initial cost and requirement of an expensive culture media, and there could be chances that the propagated plant will be less resilient towards diseases and environmental changes. So these are the disadvantage of callus culture. A modification of callus culture is cell suspension culture. When callus pieces are agitated in a liquid medium, they first dissociate and then reassociate in that particular agitation settings. And this is better for nutrient supply. Continuous agitation makes sure the nutrient is reaching even in the deep layer of the callus. But while in a stationary condition, in a solid medium, this nutrient might not reach into the inner part of the callus, and that might lead to the death of the callus in an immature state. And this happens a lot while culturing callus. So again, we start with the explant source. We induce a callus growth, and once callus has grown to a particular stage, we would take the callus and put it in a stirred tank reactor, where the callus would be stirred and the nutrients can reach deep inside the callus, and this is known as cell suspension culture. Cell suspension culture has a lot of usage. Let me introduce you to the usage of that. So, cell suspension culture is used widely to produce secondary metabolites of plants. Secondary metabolite of plants might have medical importance. So there are two types of metabolites in plants: primary metabolism, which is respiration, photosynthesis, and other biochemical pathways. And secondary metabolism involves phenolics biosynthesis, alkaloid biosynthesis, or flavonoid biosynthesis, etc. So these compounds, phenolics, alkaloids, and flavonoids, they might have medical importance or relevant for human benefit. For example, paclitaxel, which is derived from a particular plant, has anti-cancerous property, whereas digoxin is used for cardiac treatment or treatment of cardiac arrhythmia. Diosgenin, which mimics a steroid-like molecule, is important for birth control pills. So it has an anti-fertility effect. So we understand that secondary metabolites can be grown with suspension cultures and its usage in terms of human benefits. Now come to protoplast culture. Protoplast can be generated by stripping off the cell wall of the plant cell. So what is protoplast? Protoplast is simply a plant cell which is devoid of its cell wall. And there are certain benefits of protoplast culture. Let me tell you that. So protoplasts are widely used for genetic manipulation in plants because incorporating a genomic DNA or a plasmid DNA into a plant cell is very difficult due to the cell wall. Once the cell wall is removed, it is kind of easy to get the DNA of your choice inside this plant cell. So protoplast preparations are actually used by plant scientists for genetic manipulation. Other than that, there are many techniques known as protoplast fusion, which are used to overcome species barrier. For example, a species of tomato and a potato are different, so they cannot reproduce with each other, right? Because there is a species barrier, and that can be overcome with protoplast fusion. So protoplast from one plant is prepared and another plant is prepared, and then they can be fused with polyethylene glycol treatment. That leads to these fused cell types. And this is called as somatic hybrid. One kind of application is hybrids. Hybrid stands for cytoplasmic hybrids, where two cells are taken and one cell is generally enucleated. And then they are fused together. So the nucleus or the genetic material is completely coming from the other cell, whereas the cytoplasmic components are mixed up between these two cells. Ultimately, they can give rise to seedlings, and these are known as hybrid variants. And hybridization technique is used for plant advancement or plant tissue culture advancements. So let me talk about the advantages of this technique. First of all, it can overcome sexual incompatibility barrier between two different species, and it can be used to study cytoplasmic or mitochondrial genes and their inheritance and their importance in terms of plant growth and other aspects. Let's talk about the embryo culture. The embryo of different developmental stages formed within the female gametophyte via sexual process can be isolated and cultured as a whole under aseptic condition, and this is known as the embryo culture. There are certain advantages of embryo culture, such as like overcoming the embryo abortion due to incompatibility barriers, overcoming seed dormancy or self-sterility of seeds, shortening of the breeding cycle. All of these are advantages of embryo culture. So we pretty much looked at several plant tissue culture techniques. Lastly, we would talk about hydroponics. Hydroponics is a method by which plants can be grown in a soil-free situation.